Hi, my name is Kayla Latoza, and I am here with Divonne Franklin, and we've been doing research with Dr. Nicholson on the potential carcinogenic effects of red dye 40 on yeast cells and Drosophila model organisms. Our main focus was cancer. Cancer is a disease characterized by abnormal, uncontrolled cell division and is among the leading causes of death worldwide. In 2018 alone, according to the National Cancer Institute, there were 18.1 million new cases and 9.5 million cancer-related deaths worldwide. This is due to the exposure to environmental factors such as preservatives and food dyes, such as red dye number 3, have been implicated as potential contributing factors leading to cancer, making red dye number 40 a major topic of interest for our research. Unfortunately, repercussions of red dye number 40 exposure, as well as long-term effects, are still unknown. The fruit fly, Drosophila melanogaster, and yeast cells can be used as model organisms in which to study cancer, as fruit flies and yeast cells are homologous to humans, show less redundancy, which means fewer duplicate genes, and a large proportion of genes that are responsible for human diseases have the same homologs in both flies and yeast, including genes that cause cancer. Fruit flies and yeast cells are amazing model organisms because they are cost-effective, have short gestation properties, which means we can receive a lot of data in the short amount of time, since Divonne and I only have one semester for research. Examining the effects of red dye number 40 on cellular growth in yeast and fruit flies is an accurate way of determining whether it might have carcinogenic effects in humans. This study was performed to test the effects of red dye number 40 in both Hawaiian punch and red dye 40 on its own on cell growth to determine whether this specific dye might be carcinogenic. The first thing that we tested was the initial viability and growth studies in fruit flies. We first tested the effects of red dye number 40 on fruit flies by rearing fruit flies on 3 different sets of cornmeal agar food containing H2O, which was the control standard for fly food, 100% Hawaiian punch, and a combination of 50% water and 50% Hawaiian punch. With the 3 different concentrations, we have 3 test tubes per concentration. We also used 3 replicates of 10 adult flies that were maintained on each food for at least 1 week at 25 degrees Celsius. To test, the second test that we did was test for effects at different life cycle stages. 3 replicates of 10 larvae were also reared on each food type. As a result of these tests, we created a graph on the left that presents the averages of the number of fruit flies that survived in the different concentrations of fruit punch. You can see that the fruit flies had a better survival rate in the 0% concentration than the flies that were in the 100% concentration. The bar graph on the right displays the averages of the numbers of larvae that were able to pupae and fully develop into flies. We noticed that most fruit flies developed fully in the 50% concentration, but there was not a huge significant difference of development in any of the 3 concentrations. Therefore, we could not determine that the levels of the red dye number 40 in the Hawaiian punch alone is carcinogenic. Now, Divonne will discuss the growth studies in yeast.
Thank you, Kayla. Hi, I'm Divonne. And after a couple weeks of testing and observing the Hawaiian punch with the fruit flies and not getting the exact data we wanted, we decided to specifically test the red dye number 40 that is found in the Hawaiian punch. So we used yeast cells to observe any uncontrollable cell growth. We first grew yeast cells on YED plates containing different percentages of red dye number 40. Those were 5%, which has been implicated in cancer in rats, 2.5%, 0.5%, 0.25%, 0.05%, and 0.025%. As you can see in figure 4, the YED plates with the different concentrations of the dye that also gave the plates a nice red gradient color. We then incubated the plates for 48 hours at 30 degrees Celsius, and we ended up not achieving the results we were looking for after growing the plates and counting the colonies on each of the plates. So we decided to grow single yeast colonies overnight in YED and YPD liquid cultures with the same percentages of red dye number 40 to spread on the YED and YPD plates. Before spreading the yeast on the plates, serodilutions was performed to achieve a countable number of cells. We then made 3 sets of plates for each percentage of red dye number 40 and incubated those plates for 48 hours at 30 degrees Celsius. After incubation, we counted the number of colonies. As you can see in figure 5, the amount of colonies that grew on the different media. And after counting, we saw a general trend, but nothing that was an indication of cancer. In figure 3 is the line graph that shows the averages of the number of colonies that were spread across the 3 plates for each percentage of the dye. As you can see, going along the line of 0.025% and 5% of the red dye concentration, there seems to be a slight decrease of cell growth. However, these results were not drastic in the cell growth, but there may be an indication that cell death had occurred in the higher concentrations of the dye. Overall, in our research, there was no observed drastic changes that occurred with the fruit flies and no drastic observed differences between the deaths of the fruit flies with Hawaiian punch in their food and those without the added punch. However, in the spread plates, there was an indication that the red dye may not be good for your health at high concentrations, but it was not a sign that the dye was carcinogenic. At the more concentrated levels, there was less cell growth, which may indicate that the red dye was killing the yeast cells, whereas the lower percentage of red dye had more cell growth. This study is still being investigated, and the next direction will be performing serodilutions to get a smaller number of colonies to confirm that those differences that we saw earlier were accurate with the different concentrations of red dye. In conclusion, we are excited to continue this project for any further data of the correlation between the dye and the cell growth. Thank you.